recording that. Yep. All right, I guess we'll get started. There's just a few of us now, but other people might come on. I don't have the chat up. I suppose I'll be able to see a little notification. So if you have a question, though, just unmute yourself and go ahead and ask. I should be able to hear you. So, all right, I'm sharing the board, and if you can do this however you want, you can, resize me and make me big and look at the board on your own Miro board, or you can look at the shared screen through the WebEx, however you like to, want to do that. But this is on the board, the same Unit 5. And so what I put here just to show you is the PDF that's online called the Final Review. Basically, it's just the major, for each chapter, the learning outcomes that we had before, but it's, extracted all into one document, and there's some figures and things like that and tables and stuff. So I have this all as a PDF for downloading, but then I've also extracted those pages as well. So you could just look at all of them, laid out, and I put arrows because it was a little bit, it just goes across, snaking down. Okay. And then I also have this PDF here, which is these review graphics slides. And I've extracted those. We'll look at some of them today, but they might be, if you want to look at those later, you can do that as well. And then we'll write some stuff. And then I also have this board that is, I call it Review Practice to Copy. So basically there are a few things here. One is this little concept map about microevolution stuff, and maybe we'll fill this out together, fill it out together. I don't know. I wanted you guys to be able to use it as practice, to make sure that you could complete it. So it's all the microevolution ideas. Of course, you can make these kind of concept maps for anything: macroevolution, plants, red algae, whatever, where you're just branching out stuff. And of course, I use this with just the mind mapping tool in here. You can do it by hand. And then I have a chart that's going to tell you the most important individual organisms that you know, because you don't have to know a whole lot of individual organisms, but these are some of them. They're mostly ones that give diseases or sickness. And so you can see I've told you whether the bacterium or a protist, and here's the facts, and you'll sort those into this table. And then there's some charts here where there's different organisms and some options with overlapping. So it's like a Venn diagram. It's just not like it's, squares, rectangles instead of circles. But you'd sort these guys into like green algae. Is it unicellular, colonial, multicellular, or any combination thereof, different green algae. So then we've got one for algae cellularity, pigments, to remind you about that, cell wall materials. And it's not just the protists anymore. Now we're including the, small, there we go. Now we're including fungi because we've gone through those, and we're including land plants and all that kind of stuff. So, and this one, structures for mobility. Animals, and this doesn't have to be sorted. obviously, because animals, there's multiple mobility. I'm just saying the things in orange are animals, the things in green are plants. And then there's just this little thing, monocots, eudicots, to remind you of that. And you can sort these pictures into there. And then the 2 descriptors, because you'll have to do that sort of thing. So we might do that a little bit later. If not, or if you want to practice it again later, of course, you can just copy it. So don't mess up with this original one. What you want to do is click on the title, and then you can either use the dots and do copy, not link, but here, copy, or you can do Control-Command-C, and then just find somewhere else on the board to put it, and you can paste your little review board over there. You can name it your name, and then you can work on that. And I have the answer keys here, so when you're finished, you can move and then put back the yellow thing to see if you were on the right track or not. So you just put this board somewhere, that's out of the way of the other stuff. And yeah, like I said, name it your thing so people know not to mess with your stuff. You can even put a big sticky on it over here or something like that. I'll just leave that one copy since someone might want it. But yeah, let's go to talking about some of the concepts. So someone wanted to review plant life cycle, which we definitely will. I guess we could start with that. I mean, I was going to do some Hardy-Weinberg problems to remind you of that. There is ecology in the sense of things like this that we've talked about the whole semester, like nutritional modes, photoheterotroph, chemoautotroph, photoheterotroph, photoautotroph, chemoautotroph, photoheterotroph, chemoheterotroph, and then the lithotroph, that one in there as well. So that idea, the ecological roles of like what's a symbiont, what's mutualistic, parasitic, commensalistic, ecology in the sense of this, where it's like primary producer roles that we've talked about, who is a decomposer, that sort of thing. But we don't have a lot of stuff like in those last chapters. It's just the things that we've gone over, kind of brought together. So just to let you know about that, you don't have to worry about that last ecology PowerPoint in particular. Okay. Is there anything else that people had thought about that they wanted to make sure that we covered? Of course, you'll need to know phylogenies. Phylogenies, life of all the groups that we've talked about, life cycles. The names for things. Okay. I know people tend to feel pretty comfortable with the evolution stuff, but it was a little while ago. So, I often start with that, but I didn't know if you guys felt like it would be helpful or not. Or you still feel really comfortable with evolution. Can I get some feedback there?
I think going over evolution a little bit would be good.
A little bit, since it's been a couple months now.
Yeah.
Okay. So when we look at our review sheet here, here's just ideas about evolution. Then you got the phylogenetics, so interpreting trees. So I could show you a couple different types of trees, and you'd have to tell me what they say. And remember, you're pretty good now about not over-interpreting trees, but they're showing the relationships between the groups. Then we've got evolution of populations, which include the Hardy-Weinberg problems, as well as the 4 mechanisms of microevolution, how variation comes into the population, how natural selection works, that kind of stuff. And then you got speciation, about the types of barriers, about allopatric versus sympatric, how those things would work, what adaptive radiation is, that sort of thing. And then we get to patterns of evolution, which are, there's a few different things in the timeline. And then, kind of like the order of things arising on Earth. And the idea from the very beginning, when we did like the what Darwin never knew, which I have a little thing to show you about like changes in the body plan genes regulate the switches differently, and then those can regulate the stuffed genes differently, or make stuffed genes express differently, and then that can lead to, you know, differences that then can be selected on. So, okay, where do I want to start here? These are not. So let's jump to. Okay, we'll just start with this. So, to remind you, in order for any kind of evolution to occur, there has to be variation in the population, right? Because if everything is clonal or the same, which some populations are, but if it's all the same and it stays the same, then there can't be any evolution because there's nothing to shift to. There's nowhere to move, you know what I mean? Like, so it's important to understand the 5 ways that we can get genetic variation in the population. So the most basic one is are mutations, so changes in the DNA sequence, as you know. So that can change the protein coding genes, so actual proteins, or it can change regulation. I guess we can write that down. So mutations could be in protein coding genes. It could be in the switches, mutations in the switches, which are regulatory regions. It could be regulatory RNA genes, genes that code for regulatory RNA. So mutations in any of those can then give changes in the outcome. Altering gene number and position. So this is that thing where you have one copy of the gene and then it accidentally gets duplicated in the genome. And so now you have 2 copies, and then by itself, that's probably not going to do much. But if one of those copies starts accumulating changes in it, mutations happening, then that other copy may end up having a new function. So this is the way we get all of our smell receptors, because they're all very, very similar to each other. They're just the gene just got copied a bunch of times and diverged. So then we have like slight tweaks on the same protein, like little bits of different versions of it. And that allows us to smell different, detect different chemicals by smell. So this is that altering gene number or position. Or if you have like genes that when they're getting copied during mitosis, like they accidentally get inverted in the new DNA, then of course that'll change the sequence. So it's a little bit larger scale changes. Sexual reproduction. Now there has to be some variation in the population to start with for sexual reproduction to do anything. If it's clonal, then even if it's sexually reproducing, everything's the same, like genetically, so it's not going to create variation. But if there's variation in the population, sexual reproduction, of course, shuffles the genetics in the offspring. So that's not only because there's 2 parents, so each of them give, some of their genes and it mixes the genes from the 2 parents. But we also know that sexual reproduction is often involves meiosis, which has the process of crossing over in it. So that's shuffling combinations of genes to start with. And it also has the independent assortment during the process, which makes you end up having lots and lots and lots of different types of gametes with lots of combos. And then when the 2 parents donate 1 gamete each, then it's even more combinations. So that's sexual reproduction generating variation. Gene flow generates variation only if it's going into the population. Because if you're having, remember gene flow is when you have individuals or gametes or spores, something like that, coming into or out of a population. But for genetic variation, it's only when it's going in, because otherwise, you might be losing variation, or it's not making anything different. And it's not just into It's into the population with new alleles or completely new genes, but often new alleles. So it's like you have a population of brown beetles and green beetles, and then all of a sudden you have a new beetle come into the population, same species, so that it could mate, but it brings along some of that same gene, but it's a version that makes it yellow. So now you have a new yellow allele in the population, which now gets mixed into the, to the group. Whether that's going to become common or not depends whether it's selected for, because if you have one copy of the gene come in, the population's large, could just get lost. But it does bring the variation in, the gene flow, only in 2 for this one. And then we can have lateral and horizontal gene transfer. So those 3 types I'll show in a second for endosymbiosis or for bacteria. This also happens in plants, transferring genes. The thing here is that it's like for these guys, it's a direct change in the living organism's DNA. So this is where it's picking up DNA from another source, and then it's a single cell, in the bacteria at least. And so it picks up DNA from another source, incorporates that into its own genome, and now it has new characteristics. Or it picks up a plasmid DNA and then incorporates it, and then it just stays in the cell and it has new characteristics. Of course, endosymbiosis, which was that engulfing that led to the organelles, creates genetic variation because now there's like literally other organisms living inside the original organism. It's really creating organelles, and then they bring along with the genes. So let's remind ourselves about these types of gene transfer, though, and this is both for genetic variation as well as part of the bacteria chapter. So we have the 3 processes of genetic recombination in bacteria or horizontal gene transfer, and you wrote about these in one of the essays. So we've got transformation. So that's basically when you have those bacteria, and they're the same type, same species, let's say, are very closely related, maybe. And one of them dies, and the DNA fragments are out in the environment. Then one of the living ones can sometimes pick up a piece of that DNA from the environment. And then if that DNA is homologous with the DNA inside of that bacterium, like it has, similar sequence, it could be swapped into the genome. And then if that is the case, and it repairs the other now mismatched DNA with the donor DNA sequence, using that as a template, then you have a new little bit of DNA in the bacteria genome, the recipient's genome. So now it might have some new alleles on there, different versions of the genes than it used to. So you can see this is different because it's the actual blue bacterium here that is now still living itself as the blue bacterium, but now it has new genes in it. So that's transformation from the environment. We can go over to transduction, which is when you have that accidental gene transfer through a phage, a bacteriophage, the virus. Let's see if this will. Oh, I'm just sharing Miro. Oh, it does work. I found it. Oh. Okay, let's see if I can. Share something additional. Okay, so can you see the Chrome window now where the virus is moving?
Yeah.
Great. So this is the transformation process, and some people had some problems with this on the exam. So basically you have this virus, and the virus is one that infects bacteria, the phage type. So it's going to insert its DNA into the bacteria, which is how phages work. So that's viral DNA in green. And that's going to induce the chopping up of the bacteriophage genome. Now, this is the, let's see, will this work for me? Yeah, this one right here is the bacteria or the phage that we are concerned about. So usually what's supposed to happen is the bacteria DNA gets chopped up and just goes away. The phage DNA, which it's forced the cell to make, is what gets incorporated in the new phages, and then it's all just virus DNA. So this green stuff is virus DNA. And that's the normal scenario. But occasionally, accidentally, some of the bacteria DNA from that host will get packaged, potentially. And so this is the funny situation, the mistake where the bacteria DNA is in there. And not everybody always seems to grasp that part. So you've got this bacteria DNA inside the virus now. Then the virus can lyse. Most of those phages with the virus DNA is just going to go infect other bacteria and, you know, put the virus DNA in there, and it'll just be like a viral infection. But if you have one that has the bacteria DNA in it instead, and it's being accidentally carried by the virus, then it can put the bacteria DNA in there. It's not going to cause the chopping up of the genome because it's not virus DNA now. It's just bacteria DNA. And so if it's homologous, then it can do the recombination there. And then you'll have the donor DNA from that bacterium that was killed. Now it ends up in this new bacterium. And then this little piece that got switched out will get chopped up. And so now you have potentially new alleles in this recipient bacteria's genome. So that's transduction. And then the last one is conjugation, which is the direct transfer from one bacterium to another. Again, this is quite different than sex. I know people call it bacteria sex, they call it sex pilus, but I really think that's misleading, because sex is when you have two parents creating a new organism, right? This is not. This is just two bacteria. One donates a copy of some of its material to the other one, and then it, you know, has new characteristics potentially, but then they just still exist on their own. So you don't have a new baby bacterium. That's not how this is working. You're just transferring between those. Oops. So if we look at that here, there's a number of situations, but one situation is where you have, there's the genome, the chromosome genome on the left, and then here's a plasmid, which has fewer genes on it. It's one of those circular pieces of DNA in the bacterium. You can see this donor bacterium has the ability to make a conjugation pilus. So it'll be able to stick to and make a hole in other bacteria. So there's the conjugation pilus. And so here's the recipient bacterium. And if you have the donor bacterium attached to it and it pulls it close and forms a little bridge between the two, like a little tunnel in between the two of them, you could get a copy of, like you have one copy of the plasmid get fed into the other bacterium. So notice it's not transferring the whole plasmid, it's just transferring one strand. Then what you'd have is each part here now use the remaining, or use the DNA to make the new strand, because it has to be double-stranded. And so both of them now have that plasmid in there. And in this case, it passed on, part of the plasmid was the ability to make this conjugation pilus. So then it has a new characteristic now, because now it has the I guess this would probably be an F plasmid, the fertility factor plasmid, which would allow it to make the conjugation pilus. Now there's lots of different versions of this, but this is kind of like a basic one. And here's another thing. So you have the conjugation or F factor plasmid, but let's say you have another plasmid in here. Maybe this one's an R plasmid, which is for, like it has genes on it that can allow bacteria to be antibiotic resistant. Or you might have a plasmid that has genes on it that allows the bacteria to use a certain amino acid for food. So, there's all kinds of things that could be on the genes, but the same sort of situation happens. In this case, you're going to have one copy of this other plasmid go in there. And then each part will make the other strand. And now it didn't get the F plasmid transferred, so this one can't pass it on. But now whatever characteristics the genes on this mobilizable plasmid confer, now this like little bacterium on the right is going to have those genes now, or have those traits. So that is conjugation. Con meaning, or co meaning together, I should say. Okay. So that's the horizontal gene transfer, last mechanism of getting variation in the population. So if you have variation, will the population evolve or not? And this just depends on what the environment and situation the organisms find themselves in. Or I should say the population finds itself in. So there's basically 4 mechanisms we talked about that can cause microevolution. And remember, that's just a shift in those allele frequency changes over time. Let me get rid of that. Okay. It's a change in allele frequencies. So basically we're just saying, is the population shifting towards certain genetics over other genetics, if you will. And also, I found out the other day that some people, and you probably know this by now after Bio 2, but just because some population's evolving doesn't mean it's speciating, right? So microevolution is usually, you still have the same species, like the mice that had the fur coat turning from dark color to light color. I mean, it's still the same species of mouse. It's just the population has evolved into a different coat color depending on the environment. You can have speciation that can result from an accumulation of microevolution mechanisms, as well as especially ones to like if they produce reproductive barriers, but in itself evolution doesn't equal speciation. Those are actually separate. Just FYI. So we have the 4 mechanisms here, and natural selection is the one that you have heard about for a long time now. So remember, this is none of these are goal driven. Natural selection does cause adaptive evolution. That just means that it's shifting when the genetic changes are shifting the population. They shift the population towards the genetics or phenotype that results in better survival in the environment. That's what I'd say. So, they're more well adapted over time. So the population becomes more well adapted to that current environment. And of course, remember, it's not just survival, it's also reproductive success. I guess we should say better survival and reproduction in the environment. Okay. So it's not just enough to survive. You need to reproduce in order to pass on the genes. Otherwise, it doesn't matter that you survived. Not in the sense of evolution, at least. Okay. So natural selection can cause microevolution because that's obviously shifting towards whatever's surviving and reproducing the most successfully in that given situation. Genetic drift. Now remember, people still get confused between flow and drift because I understand they sound a little similar, like drifting and flowing. Aren't those like the same? But genetic drift, the reason it's called drift is because if you look at like a population that has 2 phenotypes and it starts 50-50 of the phenotypes, so both phenotypes are, starting here, so that's 50% of the population. Genetic drift, even without any selection, if it's very small, so this is the one with small populations, most impacted. Sometimes you get an allele that instead of staying constant, it just sort of drifts to 0. And the other one would just sort of drift to 100%. So that's where the name drift comes from, because it's not a selection event in this case. It's just because the population's small and there's randomness in what actually happens as far as like, you know, it's not perfect reproduction numbers and that sort of thing. You can also have, you know, the founder effect. We'll talk about that in a second. And the catastrophic kind of things going on where it's random events. But the name drift actually comes from the fact that when you look at populations, sometimes one of the alleles just sort of drifts off into zero for no reason per se, except for just, you know, that it wasn't a small population and you just might lose it by, you know, by chance kind of. So I guess we can say that with this too. Both small population and chance events, that's genetic drift. The gene flow is when individuals, gametes, or spores can come in or out of the population. So in this case, they come in, they can bring variation in, but they're changing the allele frequencies because they're bringing new stuff in, or their own things in. And then if they leave, it also changes the gene frequency, right? Or the allele frequency, because they're taking stuff out of the population. So, you know, if you had a population that had like 4 brown beetles and a population of 10 and 3 of the brown beetles left, your population has shifted now because you only have 1 brown beetle in your population. So that was gene flow out of the population, and it shifted the makeup of it. And then sexual selection is another one. So remember, this one is where it's the best reproducing that is getting selected for, but it's not necessarily the best surviving, right? You might actually be quite conspicuous as like a brightly colored male bird. You do make the most babies, but you don't live as long as like not as bright males. But that doesn't matter because you made more babies that survived and reproduced in the future themselves. So sexual selection is slightly different in the fact that it's not survival that's the most important thing here. It's actually just the reproduction. And that would cause shifts in microevolution towards the trait that is favorable for that reproduction. A really important thing that I hope we know by now, but if not, let's just review that, is that all of these processes, including natural selection, occur with the variation already in the population. So remember, it's not a need, and I still do have people, like even when we were talking about bird stuff, I still have some people writing about things with like, well, the bird needs to be able to fly, so, you know, it gets the wings to help them do that. But it's really the opposite, right? The logic is that the variation exists in the population. Some of the individuals are born with wing-like structures. It might not be full wings, but they're, you know, have characteristics of a wing, probably by body plan gene mutations. So those organisms that have this little wing type structure now may be selected for if it's advantageous in that environment, and then it becomes more common, and then, you know, so it allows flight. So it's like the characteristic exists already, and then it allows some sort of, or I guess I should say the genetics exist already, which then result in some sort of characteristic which then could be selected for and become more common, rather than, well, you need some sort of characteristic and thus let's get the genetics. Because like, how would that happen, right? You can't just go and be like, man, I need a wing, or my babies need a wing, how am I going to make that happen? Just doesn't work like that, unless we're talking about biotechnology, which is not the situation for these examples. So this is just very short, but I wanted to show, this is about antibiotic resistance, about how that arises. Is this still within the Miro board? Can you still see the little video once I made it big, or is it outside of that now?
It's still there.
Great. All right, so let's just watch this. You'll notice the variation, you'll notice the natural selection. Of course, just remind you that bacteria and viruses and stuff don't actually have faces, but it's, you know, for the general public, trying to make it, you know, relatable, I guess. But this is a good review of the natural selection process.
Superbugs, how bacterial evolution can lead to antibiotic resistance. Do you ever wonder why your doctor always tells you to take the full course of antibiotics even when you no longer feel sick? It's to prevent the formation of superbugs, antibiotic-resistant bacteria. Let's see what happens when you get a stomach bug. You may have only eaten a few bad bacteria, but they grow and divide quickly, and before you know it, you may have up to 100 million infectious bacteria living in your gut. But these bacteria are not all exactly the same. Every time a bacterial cell divides, it must first make a copy of its DNA to give to the new cell. Sometimes there are mistakes in the copying process, and the new cell's DNA ends up being slightly different than the old cell's DNA. The new cell will keep passing on this mistake in its DNA, called a mutation, every time it divides. All of these mutations lead to genetic diversity in the bacteria population. Because genes are the instructions that make cells, the bacterial population now has diversity. Just like humans with slight differences in their genes have different colored hair or different tolerance for alcohol, these diverse bacteria now have different responses to antibiotics. When you take your first day of antibiotics, these drugs will kill most but not all of the bad bacteria. Thanks to genetic diversity, some of the bacteria will do a better job than others of surviving the drugs. If you stop taking your antibiotics before they kill all of the bad bacteria, you will leave only the strongest of the bunch. The bacteria that survive will keep growing and dividing. This is real-life evolution by the process of natural selection. These stronger bugs can then keep evolving, collecting more mutations, and leading to even stronger, more drug-resistant bacteria. Check out this video from the Kishoni lab at Harvard to see the evolution of antibiotic-resistant bacteria before your very eyes. And here at New Scientist Live,
All right, so that's what I wanted to point out there, that the variation exists already, and then the pressure is what selects for it. So there's antibiotic-resistant bacteria all the time, but they don't get selected for unless the antibiotics are present, right? And they're not resistant, they don't have those genetics to be resistant. They have those genetics that allow them to be resistant. So it's not, the genetics do things like have, certain shaped pumps in the cell membrane that pump certain molecules out. And then why does it make it antibiotic resistant? Well, if the molecule is the right shape, the drug, then it might be able to pump it out of the cell. So did that transporter evolve to pump the antibiotics out of the cell? No, it pumps other stuff out of the cell too. But that variation, if it's allowed to pump, if it's able to pump the bacteria, the drugs out, then it could prevent the bacteria from dying, and that's what creates the antibiotic resistance. Or maybe it has an enzyme that can break down certain things, and it turns out that enzyme can break down that type of drug that you're using, so prevents the drug from working anymore, that kind of thing. Okay, so that is an important concept to leave with, is this idea of which order of events things happen. Okay. So this is in the microevolution little concept map. You'll see here that two of the mechanisms of microevolution may result from. So we've just gone over those. And I have some information to fill out, that's already filled out, and then some other things for you to add in there. And then you have the genetic variation sources, which we just talked about those. Definition of microevolution, which we talked about that, shifts in allele frequency in the population. And then talking about the Hardy-Weinberg equation now. So we can go ahead and do just a couple Hardy-Weinberg problems. And then this last thing is conditions for no microevolution, which would be like the conditions for Hardy-Weinberg equilibrium. When is it not going to be happening? All right, so Hardy-Weinberg is an equation that you can use to test whether a population is evolving or not, basically. And if you know a population is not evolving, then you can use the Hardy-Weinberg to make predictions about the population. So the equations are only used, like you can only use this P2, PQ, Q squared, that kind of stuff. You can only use the P's and Q's when you either know the population is in Hardy-Weinberg equilibrium, then you can use the P's and Q's, or you're predicting what it would be like if it was in Hardy-Weinberg. If you don't know whether it's in Hardy-Weinberg or not, you're gonna have to have information about the real numbers, or you can't do anything with it. So for example, so this is one kind of problem I'd ask here. Is the population in Hardy-Weinberg equilibrium? So there's not too many questions on the final about this, but in this case, we don't know if it's in Hardy-Weinberg equilibrium, but we do know some actual information about the population. So this is when you use this information in order to figure out the frequencies, right? So to figure it out if it's in Hardy-Weinberg, we have to do the observed, which is reality. That's what I call it, reality. So we have the observed frequencies. So the frequency of M, the frequency of N, the frequency of the genotypes. So like, what do we actually observe in real life? And then you need to compare that to your expected if in Hardy-Weinberg equilibrium. So that's when you're using the equations. So that's when you'd use the P squared, 2PQ, etc. And if those match, the observed and the expected, then that means the population is probably in Hardy-Weinberg equilibrium. Because the whole point here is that if your population is in equilibrium, then every generation, the frequency of the alleles should stay the same. The frequency of the distribution of alleles, which means your genotype, should stay the same. So everything should just like stay basically the same. If you're having shifts, you're thinking, well, what's going on here? Why is the population shifting in one way or another? Is it because it's small and we have genetic drift effects? Is it because it's experiencing natural selection? I guess actually that's what these things are. So what kind of condition here is not being met if we see shifts in the allele frequencies or the genotype frequencies? So you're looking for no mutation. So if you start seeing shifts in the population, are there a lot of mutations happening? Random mating means that you're not having sexual selection occur because it's not like there's preferential pairings. No natural selection. So if you start to see shifts, you're wondering, is there a selection for survival and reproduction going on? This extremely large population size, you're wondering, like, is there genetic drift effects happening? If a large population size would prevent those from being very impactful. And then you don't want to have gene flow. So if individuals are moving in or out of the population, that shifts your population frequencies. So you would look and say, well, if my population's really shifting, is that because of a lot of individuals are just like leaving or coming in? Is that what's happening? Okay. So that's the Hardy-Weinberg. So let's do this problem. All right, so you have blood type, MN blood type. I know we talk about ABO a lot, but you have MNs as well. Because our red blood cells have lots of different tags on them. And here's the numbers. And so this one, in order to do the observed, you have to have the numbers about the heterozygotes, which they do give you here, this MN combination. So what do we want to figure out? We want to figure out the frequencies of, we're really looking for the frequency of the alleles. We want to know the frequency of M and the frequency of N. So in the gene pool, the pool of genes, that's just like all the organisms in the group. How many of that gene is going to be the M version and how many of that gene is going to be the N version, or what proportion of the genes will be the M or the N. So in order to find the frequency of anything, you just take the number of that thing divided by the total number in the population. So like the number, if you had, a drawer of socks that were blue and black, you want to know the frequency of blue, you just count the number of blue socks and divide it by the total number of socks. So that's all we're going to do here. We're going to count the number of M alleles divided by the total number of alleles. That's going to give us the frequency of M. So the question is, how do you find the number of M alleles? Well, you're going to look at the genotypes and how many people have those. So for the MM genotype, you have 490 people with that. You'll notice that we're not looking at just MM, we're looking at the number of M alleles. So for each MM individual, they have 2 copies of that. So they donate 2 alleles of the M into the gene pool. So 2 copies from the MM individuals. So there's 490 people giving 2 copies of the M's into the pool. And then you also can get M's from these 420 people that are heterozygous. So you have 1 copy of M from 420 people. And then we want the total number of alleles. So if you look at everybody, there's 1,000 people. And how many alleles does each person have? 2. So 2 alleles from every person for this gene. And so you can work that out. I should have this somewhere. Because I probably do. Okay, 0.7. Okay, 0.7. So that means 70% of the alleles in the population for this gene are the M version, because the M and the N are 2 versions of the same gene. Now, we can't assume that 0.3 is the rest of them. It should be, but we need to use our numbers to calculate to make sure. So let's find the frequency of N now. So we're going to say the number of N alleles over the total number of alleles. So for the N, we have these individuals here with 2 copies of the N. So there's 2 copies coming from 90 people plus 1 copy coming from 420 people. Everybody has 2 copies and there's 1,000 people. So this should 9, 18, 8, yeah. Let's just make sure. Oh, that's wrong. Plus 420 divided by 1. Yep, good. It gives you 0.3, which is what we were expecting, but we just need to make sure. So that means 30% of the gene pool is the N version of the gene. So now we have the reality, and we want to know, given this reality, what do we expect the distribution to be? And then we'll see, is that what we see the distribution as? This is when we're going to use our P's and Q's. So we just calculated P. We'll say P is M. Given that P is 0.07, and then we'll call Q N, these are not recessive. Actually, they're codominant, so there's not recessive dominant, but usually your dominant's P and your recessive is Q. But if we say, given these things, that P is 0.7 and Q is 0.3, what we're asking is what genotype frequencies do we expect if it's in Hardy-Weinberg equilibrium? So for MM, if you just think about it, each M is a P, right? That's the probability of getting each M, and you have 2 of them. So in order to do a probability of 2 things happening, you multiply it. So P times P, which is, P squared, that's the probability of getting 2 M's. So that's your P squared value here. So P squared equals 0.7 squared. Right. What's the probability of getting 2 N's? So if you have 2 N's, we said N is Q. So that's Q times Q. So that's where we get our Q squared from in the equation. So that's 0.3 squared, 0.09. And then we have MN, and this is a P and a Q, right? P and Q, but you'll also remember that we could also get the same genotype NM. So it could, you know, basically it's just saying mom could give you the M or dad could give you the M, and then the other one gives you the other one. So there's two ways to get this. You don't have to leave. Oh, yeah, okay, she says that. Okay, great. So there's two ways to get that. You can either get the M from mom or the N from dad. So that's why this is P times Q. And then there's another, in this case, like Q times P. And both of these are enough to get you the combo. So you actually add them together, which is what gives you 2PQ. That's where that comes from. There's two ways to get the MN genotype, the heterozygote. So that's our 2PQ, which is 2 times P times Q, which should be 0.042, I believe. 73, yeah. Okay, so that's what we expect our frequencies to be. And then we can go and look at what our frequencies are. And this one's easy because our frequencies here are just out of 1,000. So what's the frequency of an MM person? 490 people out of 1,000. That's easy to divide there. That's 0.49. What's the frequency of the MN in reality? 420 out of 1,000. And then 90 out of 1,000. So these are the reality, the observed genotype frequencies. And then we say, does that match? And if you look here, 0.49, 0.42, 0.09, those do match what we thought they should be, given the frequencies of the P and the Q. So in this case, we'd say, yes, the population is likely Hardy-Weinberg equilibrium because the frequencies that we see, like the distribution of the frequencies, or the distribution of the alleles is as expected. You can also tell if it's in Hardy-Weinberg or not if you track over a number of generations and you look at the frequency and you see is it changing or not. So, you know, I'm not going to do that for calculation purposes, but that's another way to see it. Or I could tell you something though that was like the frequency is this in this generation and this and this and this, and if it's changing, then it's not in Hardy-Weinberg equilibrium. So that's one type of problem that you'd have. The other type of Hardy-Weinberg problem that you have is just using the equations, basically. So a problem like this, where I tell you it's in Hardy-Weinberg equilibrium. So cystic fibrosis is a recessive condition that affects 1 in 2,500 babies in the Caucasian population of the United States. What's the carrier frequency in the Caucasian population? Okay, so this case, and what I have to tell you, the US population is basically in Hardy-Weinberg equilibrium because it's really big. There's not really selection going on. Not particular sexual selection either. We do have some drift, people leaving and coming, but the population's so big, it's not like you have like, you know, massive amount of people leaving and no longer being in the population. So it's basically Hardy-Weinberg equilibrium. So what does this mean? When I tell you that, it means you can use your P's and Q's right away. So what are they asking for here? Carrier frequency, that's the combo genetics, of course, right? The, like, we'll say big A, little a, or in that last case, MN, but here we'll use big A, little a. So what's the carrier frequency? Remember, the combo one is 2PQ, the combination, the heterozygote. Because remember, carriers are heterozygote. That's what carrier means, that you're carrying the little a. So you got 2PQ, well, that's what we're looking for, which means that we're going to need to get a P and a Q somehow to multiply them together. So the best thing to do is your key here is solve for Q. You're looking for Q. And really, usually you'd probably find Q squared. Solve for Q, the recessive. Okay, so what does it say? This says cystic fibrosis is a recessive condition. So that means that if you have CF, you're little a. And they tell us 1 in 2,500 babies has this. So that gives us a frequency there. So what is Q, what is this telling us? 1 in 2,500 babies, that's a frequency, is little a, having cystic fibrosis. Little a is Q squared, right? Because it's Q times Q again. So what they've given us is the frequency of the homozygous recessive genotype. So 1 out of 2,500. Now, if we said that, you know, we gave you the dominant feature, saying like, you know, I don't know, this would be a silly, because it's 1 out of 2,500. That would be silly to put, but like, let's see here. In this case, let's say, okay, there's brown is dominant over the color white. Instead of giving you 40% of all butterflies are white, let's say I told you 60% of all butterflies are brown. What are you going to do? Find the recessive. So if white is the recessive, don't use the 60%, go and use the 40%. So you're going to always try to get the recessive, because there's only one way to be recessive. There's only one way to get cystic fibrosis. Little a. There's two ways to not have cystic fibrosis. Big A, or big A, little a. So you can't use P's or PQ to solve for it, because like the dominant is all the same. You can't tell the difference. So find the recessive, find out what the recessive is, find out the frequency of that recessive, so Q squared, and then you can solve for Q. So basically here you just take the square root of this, 20 square root of 2,500. Is that easy to do? Should I know that? Okay, so Q is 0.02. Yeah, because 1/5, right? Plus one more, 0. Yeah. Okay, so that's Q squared, which means now we can find P, because P is just the rest of it, right? Because P plus Q equals 1. All that means is that you have the frequency of the big A allele and the frequency of the little a allele give you 100% of all the alleles, right? Which is what 1 is. So if Q is 0.2, then P is the rest of it, 0.98, which is just saying 2% of the alleles in the population are little a, cystic fibrosis alleles, and 98% are not. And then what are we doing? We're solving for the heterozygotes. So we'll do 2 times P times Q. .0392. So what's the, this is the frequency of carriers in the population. So you'd expect about 4% of the population to be carriers, is what this is saying. Yeah. That's your answer. The frequency of carriers is 0.0392. Okay. So the key here is to find Q squared, square root it to get Q, then solve for what you need to solve for. Okay. All right, so moving to speciation, allopatric, sympatric, like the barriers. Remember, for any speciation, like the groups start to diverge for whatever reasons. There's lots of different reasons, right? That they're either separated geographically, in some way there's no gene flow. Sympatrically, there's just no gene flow because of other, not geographic separation, but because of other preferences. And then you get these changes in the separated populations because their genes are no longer mixing. They change in different ways. And what has to happen, though, the key thing here is this stuff that's changing differently in the populations must lead to a reproductive barrier. If it does not lead to a reproductive barrier arising, then the species, when they come back together, they, like the diverging groups, they'll just fuse back into the same species because they'll be able to still breed. So the deal is that the changes that are going on in the populations while they're separate must lead to one of these reproductive barriers here, whether it's pre or post-zygotic, and that keeps the population separated when they come back together. So you just want to make sure you go over these types of barriers. Anything with hybrid in it is going to be post-zygotic, because hybrid means that you had a baby, or at least a fertilization event that's a hybrid, a mix of the two species. But then for some reason it doesn't work. Whether it's that organism that's sick, which is a reduced hybrid viability, or just dies and is never born. Reduced hybrid fertility, the organism could be really healthy, but it just can't reproduce itself. The hybrid can't. Or hybrid breakdown, meaning that the hybrid's okay, but then when the hybrid starts to mate with other hybrids, then those offspring of the hybrids are not, at least some generations, they don't do very well. They're probably not viable or not fertile. So it's, the hybrid itself is all right, and the hybrid is fertile, but the offspring of hybrid crosses doesn't go very well. So that's the hybrid breakdown. And here are some examples here, just to remind you. Okay. Let's do the life cycles, since someone asked specifically about that, and I want to make sure we do get to it. Okay, so there's 3 types of life cycles that you need to know. The animal, the plant, and the fungi. So we can start with plant, doesn't matter. So the plant life cycle has 2, it's called alternation of generations, because one generation is a gametophyte, the next generation is a sporophyte. They may be separate plants, or they may be nested inside each other, depends on the type of plant it is, like which group it belongs to, but it's an alternation between a multicellular haploid and a multicellular diploid, and then a multicellular haploid and a multicellular diploid. Whereas like animals, we never are in a multicellular haploid. Our haploid cells are only our gametes for most animals. So this plant, they'll go back and forth between those. So what you need to remember is sporophyte or gametophyte. We'll just do sporophyte though, sporophyte. So sporophyte is the multicellular version of the plant that's diploid. So if you remember, it's diploid and multicellular. That's the sporophyte. In this case, this is going to be, on most things, the thing that you see. So this is going to be the daisy that you see, or the fern that you see, the conifer tree that you see, except for mosses. In mosses, the sporophyte is quite small. The part that you see of a moss, that's the gametophyte. But everything else, the sporophyte is the part that you're going to observe as the plant. So sporophytes, which are diploid, 2N, are going to make spores. And remember, you should know by this point that spores are always haploid, and they're one cell. So we have to think what process occurred here to go from a sporophyte to a spore. Well, if it's going from 2N to N, we know that that means that it halved the amount of DNA in each cell. And the process by which you divide to get half the amount of DNA in each cell is meiosis. So we made spores by meiosis here. So all plants make spores. Not all plants make pollen, only seed plants, which are the gymnosperms and angiosperms, but all plants do make pollen. Do make spores. So spores are N. So spores are going to grow up into the next generation, which is the, not sporophyte, but the gametophyte. Now this one is N, because sporophyte and gametophyte are the opposites. So sporophyte, if you know that that's diploid, then you know the gametophyte's the opposite. So that's haploid. It's multicellular, which means we needed to do some division to get there. So this is N to N. So how did the spore, which is this little cell, like fall on the ground and grow up into the gametophyte? Or it may have been nested within the pine cone and then grown into a multicellular gametophyte, or in the flower and grown into a multicellular gametophyte. How did that happen? N to N means the ploidy is the same, so we must have done mitosis here. So the spore goes mitosis to make a gametophyte. Gametophytes make gametes. Gametes, as you know, are always haploid and one cell. So again, we have a haploid to haploid. So haploid to haploid, anytime you're keeping the ploidy the same, it's always mitosis. So the gametophyte does mitosis in order to make gametes, which are eggs and sperm. The plant may be able to make both of those itself, or there may be different versions of the plant that make eggs and other versions that make sperm. So both of those are possibilities depending on the species. And then those gametes would come together. So you'd have to have an egg and a sperm. They'd make a zygote. The zygote would grow up into a little embryo. And then the embryo grows into a sporophyte. So I don't always put the embryo in there, but just to FYI. But basically it'll be a zygote and then the sporophyte. The zygote is the combination of two gametes. So if we had N and N, now the zygote's 2N. It's one cell. We've got to divide up into the sporophyte. And you'll notice that we doubled the ploidy here. So N, and then there was another N gamete over here. And that process, of course, is fertilization. And then in order to take this one little single-celled zygote up into a sporophyte, 2N to 2N, ploidy is the same. So that's mitosis. So ploidy stays the same, mitosis, ploidy gets halved, meiosis. So which are the only cells here that are genetically varied, actually? There's only one spot where you end up with genetically varied cells from the division. You can just turn on your mic. So which one of these stages has actually genetically varied cells?
The zygote and embryo?
Zygote and embryo. Well, every cell in the zygote, it's single cell. So the one cell itself in the zygote should be the same. Now, this is interesting. So you are correct in the sense that fertilization, because it comes from two parents, the gametes are going to combine and make different zygotes. That is correct, because of that sexual reproduction. So you are right that the sexual reproduction there is going to create a bunch of different types of zygotes, depending on which egg and which sperm come together, because it comes from two separate parents. Interestingly, in this case, all the gametes made by that one parent are going to actually be identical, because mitosis is not generating any variation, right? So all the eggs and there's actually just one egg, like in flowering plants and other plants. So the egg would just be, would actually be genetically the same in this case. You are right that the zygotes from sexual reproduction are going to be different. Then there's also another point where we form cells that are different from the actual type of division, where they have variation in them as well. Which of the two division types gives you varied outcomes?
Meiosis.
Yeah, meiosis. So spores, every spore that's formed, they're all going to be genetically different from each other. So that means each gametophyte that grows from that spore will be genetically different. Then those produce genetically different gametes. Now, they themselves, all their gametes are the same, but then, when they combine those, you're right, in fertilization, those come together differently. So that's the two spots, actually, where variation occurs. Fertilization from the combining together, and meiosis with spore generation, because it's the process that has the variation production in there. But you'll notice that here, gametes are not produced by meiosis. It's only in animals. So that's plants. And you can see that is what is, let's see, here. That's the version of it. This is on the bottom here is the spore phyte, and the gametophytes on the top. Excuse me. Let's do fungi next. Plants can asexually reproduce, but they're basically doing that by just breaking off parts of their body that grow up into a new plant, which is a little different than the fungi, which actually make spores that make a new fungus, asexual spores. They also make sexual spores that can make new fungi too. So they have two ways of doing the reproductive cycle through spore production for both of these. So you've got the mycelium, and remember the mycelium is always haploid. Mycelium is just the body of the fungus made of the hyphae. And the mycelium is going to, can grow structures that then can make reproductive cells called spores, and those can grow up into new fungi. If it's an asexual process that's going on, the mycelium is going to make this spore-producing structure, so it'll grow, develop a spore-producing structure. So this would be something like the conidium. You might remember looking at the penicillium in class, or the aspergillus, where it had all those balls on the end of the puff. You know, it looks sort of like a dandelion, but it was spores. So that's the spore producing structure, is not all these little spores, but this like little kind of bulb thing at the end. So that spore producing structure is also going to be haploid. So it does, the mycelium can grow this structure by mitosis. Spore producing structure is going to make spores. Spores, we know, in any organism are always haploid. That goes for plants and fungi. So spore producing structure was haploid, spores are haploid, which means that we did mitosis to make those. So all the spores here are genetically the same in asexual reproduction. So you have a bunch of single-celled spores, but all of them are the same. And then the spores can puff out or be blown out or shoot out, however they get dispersed. And those spores can land on the ground. And if it's favorable conditions, they can grow up into a new mycelium. And that would make a new fungus. The fungus would be genetically identical to the original fungus. And it's end to end. So this, again, is mitosis. And so this is the asexual cycle, because there's only one individual that's contributing DNA, like it's only one parent here, this original mycelium, and it forms identical fungi. So all the fungi that come from this will be identical to the parent fungus, which is not a bad thing, especially if conditions are stable. But if conditions aren't stable, it might be useful to have the sexual reproduction, because that can generate some new variation. So that's what we'll look at here. So we have the mycelium, and instead of making that spore-producing structure right away, what we're going to have to do is have two mycelia meet up, right? Because you need, for sexual reproduction, you need two parents, or at least, yeah, genetics from two different cells. So in this case, we're going to have two parents. So there's another mycelium from another fungus. And they're basically just going to grow near each other. Or one might, if it's like a yeast, it might like be in the air and then fall on another one. But in some way, they're going to get in close proximity to each other. And each mycelium is haploid. that cells at the ends, just the tips, are going to fuse together. So that last cell on each of these is going to fuse together. And because it's the fusion of cells, it's called plasmogamy, the fusion of the cytoplasm. So you can see that that happened here. Still two separate nuclei. That's what these dots are, nuclei. Which is why this is just the fusion of the cells. So we've got, write this in the right color. Plasmogamy, the marriage of the cells, the cytoplasm, if you will. And that gives us our heterokaryotic stage. In this case, it's dikaryotic because there's just two, but you could have, you can actually have many nuclei end up in the fused cell, but there'll all be a bunch of haploid nuclei, and half would be from the one parent and half would be from the other parent. So that's important to note. Each of these is haploid. So the heterokaryotic stage, which is something like n plus n, in this case just n plus n, might be n plus n plus n plus n plus n plus n, but in any case it's all a bunch of nuclei that are all haploid, half from one parent, half from the other. Well, that's the heterokaryotic stage. Then if we eventually want to make a new organism, we need to and the whole point here is to like get genetic variation. We need to like combine those genes together so we can mix them up. So how are we going to do that? We need to fuse the two nuclei together. So there'll be a trigger and the two nuclei will fuse, creating one diploid nucleus. That fusion of the nuclei is going to be the nuclear marriage, which is karyogamy. Karyo meaning stuff relating to the nucleus, and ogamy meaning marriage. So we have karyogamy, gives us 2n. And that thing there is your zygote. Various forms it can have, but it's going to be your diploid cell that's from the karyogamy. Now mycelia always grow from spores, so a zygote can't grow directly into mycelium. It's got to go through, the zygote's going to end up making spores in some way. So what we basically have is the zygote grows into, or grows part of it into, a spore producing structure. And this is usually not like super elaborate process, but we have the spore producing structure is then going to make spores. We know spores are haploid always. The spore producing structure, I mean, it's still part of the zygote, so it's basically diploid still. And it could be formed by division, but often it's just sort of formed by maturing of some of the cells in there. So I'm not going to write anything in between zygote and spore producing. But spore producing structure from the zygote goes 2n to n. That is important. So we're going 2n to n. So we know in that case that's halving the amount of DNA. So that makes meiosis process. So these are going to be genetically varied spores. There you go for sexual reproduction. And then those spores can grow back up into a mycelium, end to end, so that would be mitosis. When it's the spores, it could also be called germination, but it's mitosis. It's a real wonky cycle right there. Sorry, it looks so crazy, but that's a sexual reproductive cycle. So every new mycelium that comes from the sexual reproductive cycle would come from an individual spore, which would have its own genetics, and they'd all be genetically unique. Who's on the Joshua account? I don't think I just have someone sitting in. I don't know. I don't know. All right. All right. So that's the fungi. And then we've got the animal, which you pretty much know. You do want to know the stages of the development, though, the gastrula, because like these other things too have developmental stages. We just didn't talk about them. But for animals, you would want to know blastula, gastrulation, gastrula, that sort of thing. So for animals, we're going to start with the adult. Usually in animals, there's a female adult and a male adult, but not always, as you know that there's plenty of hermaphroditic animals which would have both ability to make eggs and sperm, which sometimes can self-fertilize, sometimes not. But in any case, you need an adult, or you need some formation of sperm and formation of eggs. So we've got here the male adult, or the male reproductive structure in the adult is going to produce the sperm, and in the female it's going to produce eggs. And adults are diploid in animals. Gametes, as you know, are haploids, so sperm and eggs, which means we're going 2N to N for these, so that process is meiosis. So that means that the sperm are all going to be varied and the eggs are all going to be varied from the other eggs and sperm. Different than what was in plants. Then the sperm and the egg will come together. The sperm donates its nucleus to the egg. And then through fertilization, those two nuclei will fuse within the egg, which makes it now a fertilized egg called a zygote. And zygotes are 2N, which makes sense because you had two N's fused together to make 2N. So now it's diploid. But the zygote is just one cell, and now we need it to get back to a multicellular adult. So we've got to have a bunch of, division happening in here. So the rest of this is all mitosis. And it goes through a number of developmental stages that we talked about. So the first kind of thing you'll do is the cleavage stages. That's when there's a lot of mitosis, and eventually from those cleavage stages, it's going to form the blastula. This is all 2N. The blastula is that hollow ball of cells. Where where there's a lot of potential, so they're not specified yet as to what they would become in many organisms. So we've got this blastula. Then the blastula is going to start to get like that poke inside of it, and then it's going to start rolling inside to form that tube, to form the gastrula, just like that. So this process here is gastrulation. So the early gastrula, and then by the end, the gastrula will have this is a tube here, forming inside of it. Eventually, the tube will go all the way through, at least in animals with a full gut, and you'll end up with a three-dimensional structure like this, with a tube in the middle of a ball, if you will. Gastrula, of course, is 2N. that's not the most important part now. So with gastrulation, though, we've got formation of the gut, the tissue layers. So I guess we can say differentiation. That's one really important thing. Right, the ectoderm, the endoderm, the mesoderm, if it's a triploblastic. So differentiation of germ layers. The gut tube forms. Remember, when it's forming, it's called the archenteron. In here is the blastocele. The hollow part in there. The archenteron is the forming part that'll eventually make the gut. Oh, the part that starts poking in, that first opening down here, as you know, is the blastopore. And that, right, turns into the anus in deuterostomes and turns into the mouth in protostomes, or protostomy type development. So that's early gastrula, late gastrula. The gastrula will grow into... and this is all embryonic stages, blastula, gastrula, all that stuff, will grow into either, well, I guess we go through organogenesis and just be later embryonic stages. So we're making the organs, patterning the body, all that kind of stuff. Growing in size. And then either it'll go straight into an adult. I mean, it could be like a child first, but that's like, we count as adult forms when we're born, right? Even though that we're juveniles, it's not like a caterpillar where it comes out of a caterpillar and then it's going to morph into the butterfly. So we count as an adult form, even in our, children's stages. Okay. So the later embryo stages eventually go into the adult, or as we mentioned, they could go into a larva, and then that could do metamorphosis into an adult. All this stuff is diploid. That's the animal life cycle. So those are your 3 life cycles. Just need to check. 30. Okay. All right, so we have about 20 minutes left, and so anything else you want to make sure we cover? I have these nice little tables up here with the, the basic characteristics of the groups and some examples, and of the protists to remind you. So that'll also be helpful in these little sorting things. So again, the specific ones that you'd want to know. So if you're wondering, which one of these do I need to know? These guys will be the potential ones I'd have on there. And then, the different characteristics that'll help you sort. Because I might do something where I tell you, oh, you find this organism, it has these features. What is it most likely? And you can just say, well, oh, it has fucoxanthin. I know that means it's brown algae. All of them have chlorophyll A because they get that from the primary endosymbiosis. Maybe we should draw that phylogeny again of the endosymbiosis events. That might be a good one. Unless someone else has another question they want to make sure we go over.
Yeah, I think going over the phylogenesis would be really helpful, especially like over unit 2.
Okay, yeah. So the only Yeah, the phylogeny in Unit 2 that you'd need to know, well, so the domains, eukarya, bacteria, archaea, of course, which we've drawn a lot of times, so, up to this point. So I think you're okay with that. But I think this is the most important one. I'm not going to ask you about the, like the branching within straminopiles. Like you'd want to know the diatoms and brown algae are the ones that go in straminopiles and that they're in the sarclade of eukarya. But then like to know all the nitty gritties about ciliate branching and all that, I won't ask you about those specific branches, but I will ask you to interpret a phylogeny showing the endosymbiosis events, which then incorporates a lot of the groups in there that we had as far as part of Unit 2, what we learned about. So make sure I draw this correctly. Okay, it's a little tricky. Okay, so I'm going to draw this here, and that's going to be here. And then this is going to be, and then here is going to be like that. Okay. Something along the lines of this. So we have bacteria here, archaea here, and then all these groups are going to be eukarya. So one endosymbiotic event that's really important that precedes eukarya diverging was when you got this transfer, this engulfing and not digestion. Because like we said, engulfing a bacteria is not abnormal, but not digesting it and like just keeping it in your cell and then exchanging genes with it and then it becoming dependent, that's unusual. So this first event here, this endosymbiosis of the bacterium by the sort of like it's a proto-eukaryotic cell, not quite a eukaryotic cell yet. No nucleus or anything like that yet, but it engulfs this alpha proteobacterium. Okay. Let me make this in here. Alpha proteobacteria is just one of the categories, one of the kingdoms of bacteria. So it engulfs one of those, and then that, again, doesn't get digested, switches genes with the nuclear genetic DNA. So then it's like, genetics are like linked to the host cell, and then it eventually becomes what we call the mitochondria, right? So that's the origin of the mitochondrion, which then all eukaryotes have from that single mitochondrial origin. Then, in one lineage, we have bacteria that got engulfed again, this time a cyanobacterium. And that cyanobacterium ended up doing the same thing, not getting digested, because lots of cyanobacteria used to get eaten they still get eaten all the time by other cells, but it didn't get digested, exchanged the genes, became dependent, and ended up turning into the chloroplast. And the lineage of eukaryotes that derives from this directly, like it gets its chloroplast from this primary endosymbiosis, because we should mark these, both are primary endosymbiosis events. So this is a primary endosymbiosis event, this is a primary endosymbiosis event, are the Archaeplastida eukaryotes. So you do need another phylogeny for Archaeplastida with the glaucophytes, the red algae, green algae, all that stuff. It's up. We've drawn a few times, and then it's also a version of it is over here. We grouped coleochaetophytes and stoneworts kind of together in a thing called charophytes. But this is the Archaeplastida supergroup. That's what this is. So that's where this chloroplast primary endosymbiosis, it comes from for all of those groups. So in general, the glaucophytes, the red algae, the green algae, and the land plants, or embryophytes, we can call those. That's the Archaeplastida lineage. So then you're like, well, I know that other groups like diatoms have chloroplasts. Where do they come from? Because they're not in the Archaeplastida lineage. They didn't get their chloroplast from this primary endosymbiosis. They get it from secondary endosymbiosis. So they basically get it from engulfing an archaeplastid, specifically a red alga or a green alga. So we have, along the history of time, we have sometimes when we had, let's see if we can see this color, a green alga get engulfed. So let's say like that. So green alga got engulfed, and then that ended up, instead of being a green algae, then it kind of degenerated a little bit into just being a chloroplast. Remember, sometimes they still have the nucleus there inside the chloroplast, the old nucleus. But the green algae, the chloroplast, so a group that would come from that would be like the euglenids. Come from a secondary endosymbiosis. So that's what this is. This is secondary endosymbiosis. And that's how they got their chloroplast from green algae. We also have the same thing happening for chlorarachniophytes, get it from a green algae. Then we have some groups that are going to get their chloroplast from a red algae. In fact, a lot of groups. So then there's other events where you have a red alga being the one that gets transferred. And then that ends up forming the chloroplast in the new lineage. So lineages that are like this, for example, diatoms. Dinoflagellates, brown algae, coccolithophores, which are haptophytes. So lots of groups got there from red algae. So this is just an example, diatoms. Then we have a group that had none of this going on. That's the Uniconta. Diatoms is just an example of a group that would have this kind of transfer. Same with the euglenids is just an example. It's not an entire supergroup here, the diatoms. The uniconta, though, that's a supergroup that doesn't have any of this secondary endosymbiosis. This is a secondary event. Remember, uniconta are the animals, fungi, amoebozoans, which are the lobe amoebas, the slime molds. And we'll draw this out in a second. It's the choanoflagellates. And the nuclearids. So they don't have any secondary endosymbiosis. They did have primary endosymbiosis to get this mitochondrion. And then you have other groups like Giardia and all that kind of stuff that don't have secondary endosymbiosis either. So there's other eukarya. And if I was drawing this completely, then I'd draw this red algae line and this green algae line a number of times. Then there's also another primary endosymbiosis event going into, I guess we could actually, maybe we should draw that here. This is cyanobacterium. Different one though. So because it's a bacterium, that's another primary event. And I don't know about the timing of this. I'm just showing you examples of the kinds of engulfings that could happen. I don't know if this was the last thing that happened or not, but I do believe it's after this other cyanobacterium event at least. The chloroplast, this group here is the Pollinella. So this is going to be that type of cercozoan. Cercozoan is a type of Rosario. But it has what we call like, you might hear it called a cyanell, you might hear it called a chromophore. Basically, it's a chloroplast-like structure. It's just not the same type of chloroplast that like most of the other groups have. Because it comes from a different primary endosymbiosis event. So the groups that come from, I guess this would be useful. Red algae, secondary endosymbiosis. So we've got diatoms, we've got the dinoflagellates, we have the haptophytes, which are the coccolithophores. We have the brown algae. Off the top of my head, those are the big, like a lot of the big ones. And then the green algae, or the green alga, because it would be singular, a single one that got engulfed. The euglenids, as I mentioned, and then the chlorarachniophytes. Yeah. if we wanted to think about the whole thing of eukarya, what would you want to know? Want to know oops. Oh, I guess it's time. I don't think they're closing it though. Oh, sessions will automatically end at 7:50. No. Oh, 4 minutes. All right. Well, let's see if I have this drawn. There we go. Okay. So we didn't call it excavita because of the controversy between that group really being all lumped together there. But we did talk about, and you don't need to know Diplomonas parabasilids. We didn't talk about that. But you would want to know, the euglenozoans have the euglenids in it, which are like euglena. These other guys are mostly like they have the Giardia, the Trichomonas vaginalis, the Trypanosoma. So a lot of those are kind of disease-causing organisms in there, and you can actually see some examples here as well in that table. And then you've got the Straminopiles, which is the S, the alveolates, and the R, the rhizarians, and the characteristics like that distribute those groups. I'm not going to ask you about putting branching in here, so I would expect you to know that diatoms and brown algae are part of Straminopiles, which is the sarclade, but I would not expect you to be able to sort out all the branching for that or the excavita. I do expect you to know the branching of the Archaeplastida, including the glaucophytes on here too. And then I would expect you to know the Uniconta branching. And then also, of course, within that, the branching of the animal stuff, the branching of the plant stuff. Yeah, those are the big ones. So we do have a picture here of the Uniconta fungi animals. No, actually, this is bigger than that. This is just Archaeplastida here and some of Uniconta there. So fungi and animals versus this is a charophyte in plants or grain algae in plants. So that just shows that. So there's your Archaeplastida. And look at the stuff we drew in class. I think those will be most helpful. We just did animals, so just kind of refresh on that. And if you have any specific questions that come up, you can let me know. But yeah, I would definitely suggest, though, going over and making, like I said, a copy of this stuff and seeing if you could fill out those things out and sort the table and that kind of stuff, and then just check your answers underneath the yellow things, because this will help you with that Unit 2 information about, like, what are those groups, what were those groups of protists and their characteristics. You can also go back and look at that protist table that we did. So that could also help. We filled out that, protist characteristic table back on the board. I think it was on the board for Unit 2. So, all right, I guess since we're about out of time, like 1 minute, that's what I'll end on. Well, thank you for the thumbs up, Mackenzie. Hopefully that was, gave you some idea of what we're looking at and helpful, at least to remind you at the very beginning of stuff. And sorry, go ahead.
Bye.
Oh, bye. Okay, I'll talk to you guys on Tuesday. Yeah.
Goodbye.
Bye, Trevor.